Some plants are small, some are large. However, all plants start from the same place, the seed. As the plant begins its long path to adulthood, the plant releases various hormones, many of which are released during the seedling stage. These hormones have a range of purposes. Today, we will be discussing one of the 5 types of plant growth regulators, Gibberellin.
Hello, and welcome to our stop-motion video about the gibberellin or gibberellic acid transduction pathway. The pathway starts with the synthesis of gibberellin, which occurs in plastids, the most notable of which is the chloroplast. A very unrefined form of the gibberellin is synthesized and transported to the rough ER, where it is further transformed. Gibberellin may be specialized into their final forms in the cytosol before finally leaving the cell through the cell membrane. The gibberellin is transported through the plant, through the xylem, the phloem, or is reused by the same cell. This means that gibberellins are transported in both an endocrine and autocrine manner. Gibberellins are typically synthesized in early seed development and are transported to developing parts of the plant, such as young leaves or flower buds. However, gibberellins can be synthesized throughout the entire plant, depending on the situation. In the cell, the gibberellin-specific genes that regulate plant growth are inhibited by the DELA repressor in combination with the GID1 protein in the nucleus. Gibberellin acts to counteract this inhibition. The gibberellin freely diffuses into the cell through the cell membrane and connects to the GID1 cell receptor. The GID1 changes shape from the attachment of the gibberellin. This allows the F-box protein, SCF1, to attach to the GID1. This complex then has the capability to adhere to the DELA repressor. This begins the process of polyubiquitination, which attaches ubiquitins to the DELA repressor. This marks the DELA repressor as a target for the 26S proteasome to degrade and destroy. With the absence of the DELA repressor, the GID1 protein may detach from the complex and attach to the regulatory site of the specific gibberellin genes, which allows the genes to be transcribed into mRNA. The transcribed mRNA is then transported to ribosomes in the cytoplasm, where they translate the mRNA genetic code into an amino acid chain, forming new proteins. The most common protein synthesized is the enzyme amylase. This enzyme is used by the plants to degrade starch and other organic material from the soil. This promotes increased plant growth by having an increased metabolizable sugars in the plant.